Everyone, my name is Dr. Noor Bahadar and I am PhD in molecular genetics. Currently, I am working at Northeast Normal University as a research associate. For today's talk, I have chosen my topic as the CRISPR driven point of care diagnostics. I have been involved in research based on CRISPR-Cas systems for years, and here in our lab, we have established several mouse models, zebrafish models, and hamsters based on different types of gene modification, such as gene knockout, gene knock in, or base editing, or the prime editing. Today, I have chosen the topic of diagnostic as the CRISPR as diagnostics because we would want to have more affordable system for the clinical diagnosis. The current situation of clinical diagnostics need a well established laboratory that should be equipped with heavy machinery. Besides the heavy machinery, there should have people who are well trained for all the clinical testing aspects. But this is not easy. Somewhere, some clinical laboratories are not well equipped with the machinery or they don't have well experienced people. And this results the preventable deaths, the unchecked transmission, and also the treatment resistance. According to a study published in Lancet in 2020, more than 47% people in the world don't have access to the molecular diagnostics. And even in the in this era, some people don't have access to the basic diagnostics, and for some people it is still the luxury in many parts of the world. So in this case, the point of care diagnostic remains a valuable target for any device that can be considered as a point of care diagnostic. They should follow some basic requirements, and these requirements have been established by the World Health Organization. The most important, these devices should be low cost, they should have high sensitivity, they should be specific to control the false positive or the false negative results. Common people should know how to use them. They should be easily deliverable and stable in various conditions such as harsh environments, and there shouldn't have the delivery of complex machinery, and the delivery should be easy to the remote or field settings. There are several stages for a device to pass the point of care diagnostic. The very first step is the research, then the prototype, the market introduction, the market penetration, rural health provider, then hospital and the laboratory, and ultimately it should reach to the patient. Although the CRISPR systems have been evolved as the gene editing tools, as these cartoons have been shown, the basic uh, the basic systems that the CRISPR were introduced for the gene editing. But this is not only the gene editing. CRISPR have evolved far beyond the gene modification system, and they can be used as the diagnostic purposes, such as the samples can be collected from the patient. These can be the microbial pathogens, viral infection, or even in some cases can be some mutations. The samples can be the swab, the blood, or the saliva, and then the nucleic acid should be extracted and amplified. This extracted nucleic acid should be treated with different Cas effectors such as Cas9, Cas12, 13, or Cas14, and upon their introduction to the targeted sites where the sgRNA single guided RNA will translocate these Cas9's Cas systems to the targeted region where the guide RNA should be present, which will ultimately produce fluorescence, and these fluorescence should be integrated into the strip-based devices which will be easily detectable via naked eye without heavy equipments, or these should be easily detectable via normal machines. Since the purpose of my talk is not to go deep into the details of the technical mechanism of this system, rather than the purpose of my talk is to introduce the CRISPR systems as the diagnostic engine. Here I would like to mention COVID-19 as a case study which has been established and approved from the FDA during the peak time of the COVID-19. So in this case, the collection of the specimen should be collected from the patient. It can be the blood, can be the saliva, or can even be the urine. And then the nucleic acid should be extracted and DNA should be amplified. So we will have two scenarios. In scenario 1, if the nucleic acid, the targeted nucleic acid is present, it will be cleaved and the fluorescence will be produced. It means the person is infected with the COVID-19. In scenario 2, if the person is not infected, there will be no fluorescence, and in that case we won't see any band on the strip and there is no fluorescence. CRISPR diagnostic has been used in the real world in several cases such as the HPV, the tuberculosis, the COVID-19, and even for different variants of COVID-19, for some antibiotic resistant genes, and for Zika and dengue viruses also. We should have enhanced this CRISPR-based diagnostics by applying them to the smartphone apps by integrating them into microfluidics, and even if in some rural areas or rural settings, battery powered portable heating units should support this CRISPR diagnostics. Besides, AI and cloud linked CRISPR devices, which are under development, will support this idea. The real world adoption has been established. The emergency use of authorization, as we have seen in the case of COVID 19, and the detector got the FDA approval in 2020. Clinical validation requirements should be of high standard. The regulatory framework should be of standard from need the approval from FDA and the World Health Organization. Similarly, the manufacturing and scalability challenges should be met. Clinical and public health impact of CRISPR diagnostic impact individual life in terms of rapid diagnosis. The people are getting the diagnosis very rapidly. It is decentralized testing and personalized medicine, and it can be home based and self testing. At population level, it can control the outbreak such as the COVID 19. It can help us in monitoring the antimicrobial resistance, and it can help us the cost reduction in terms of minimizing the lab burden, the repeated visits and delay. And as we have mentioned in earlier slides, that still a number of people, up to 47% of world population, that don't have access to molecular diagnostics. So in case of CRISPR diagnostics, common people will be able to have access for the clinical diagnostics. There has always been challenges to new technologies, and similar is for this CRISPR diagnostics. So the scientific and technical challenges, as well as the ethical challenges and consideration, should be considered. In nutshell, the CRISPR is not only the gene editing system, it is a powerful tool to precisely target some specific pathogens, and it can be formatted as the point of care for different pathological diseases such as the COVID 19, such as the tuberculosis, HPV, and even in some cases, some mutations. CRISPR diagnostics have proven effective in real world scenarios, as I have mentioned. There are still some technical and regulatory hurdles which should be met up. The future is modular, mobile, and equitable. CRISPR can close the diagnostic divide. It means common people will be able to have access to molecular diagnostics if we handle it wisely. I would like to acknowledge that some of the pictures used in this talk are taken from the Google, and some of the pictures have been generated with LLM with a strong human control. Since our lab is entirely working, researching with the CRISPR systems, so if anyone is interested to know more about this system or they need some support regarding the project, so here I have mentioned my email and the WhatsApp number. Thank you. Thank you to everyone.